Holly Schick, and I'm the Executive Director at Saskatchewan Seniors Mechanism. SSM is pleased to be hosting this webinar in collaboration with the Centre on Aging and Health at the University of Regina. A little piece of information for you, SSM is going to be hosting a three-part series of webinars on ageism, starting on February 25th. Watch our website closer to that date for more details about that. Today, I'm very pleased to welcome Jason Vanstone from the Saskatchewan Health Authority. He's going to talk to us about the wise use of antibiotics, and we're very pleased to have him here to talk about this important subject. There will be a time to ask some questions when he's finished his presentation. So welcome, Jason, and over to you.
Thank you very much, Holly. I really appreciate the Saskatchewan Seniors Mechanism for giving us an opportunity to talk here today and talk about this important issue. So I guess, yeah, I mean, as a little introduction to myself there, my name is Jason. I've grown up in Regina, so I'm Saskatchewan grown. I did my undergraduate here and went to Ontario to do my PhD and came back and started working with this antimicrobial stewardship program in Regina when I moved back in 2015, I guess that was 2016. And so at that point, that was something that was totally new to me. And then the more I learned about it, the more, it became this thing in my brain that was like, oh my gosh, this is really important. And we really need to do something about this and talk about antimicrobial resistance and antibiotic stewardship and how we ensure that these resources are available for a long time to come. So that's kind of what I'm going to walk through today in this talk. I've entitled it The Antibiotic Apocalypse, just because sometimes that helps get people riled up to see a very interesting presentation. So I hope you guys find it interesting. And it is an important topic. So, but I'm going to kind of walk us through that world about antibiotics and how they work and why they work and what happens when they don't work and kind of what that means for us, the future of humanity. So I will start with really what you have to start with when you want to talk about antibiotic stewardship is bacteria. And so understanding what bacteria are and how they work. And I'm not going to go into, heavy details about this, but just to get a general idea. So this, what you see on the screen is kind of a generalized bacterial cell. So bacteria are living things, just like us, except that for them, everything happens inside one cell. So they're unicellular. They're made up of just one cell. And they have to do all of the things that we have to do, and intake energy by eating things, get rid of waste. They have to be able to move around in their environment to access new food sources. They have to be able to protect themselves and do all these different things and replicate. But they do all that within one cell. So in order to protect themselves, they've got several different structures that could be available to different bacteria. So you can see here it's outlined a plasma membrane, a cell wall. Some bacteria have these capsules as well. And so this is just kind of like a shell. You can think of a shell that surrounds those cells and protects all the gooey inside stuff from all the bad stuff on the outside. So very similar to our skin and our bones that protect our organs and things like that. Bacteria also have to, as I said, be able to get around in their environment. So this big green tail that you kind of see sticking out the end is called a flagellum. And it's kind of like a little boat propeller. So it spins around and that helps the bacteria swim around through, whatever liquid or sewage or wherever it's living. And then these little pili that you can see over here too outlined, those are also important for helping the bacteria to sort of stick to things and get some traction as it moves around in the environment. So bacterial cells have to grow and they have to make proteins in order to do that. So if they want to build cell walls, those are made of proteins. And so they need to build up the proteins. If they want to digest their food, they need enzymes to help them do that. And those enzymes are proteins. And so in order to make those proteins, you can see these ribosomes, they're called, and these little blue spheres that are kind of dotted around the cell. And you can think of these, I put the picture of the little machine arm there because you can think of these like little biological machines. So these are the machines that kind of read through the DNA and they direct the building of those proteins. And as I mentioned, DNA, so every living thing has to be able to replicate to make more living things. And so that the molecule that's most important there is the DNA. And so bacteria have DNA just like humans do. This big spaghetti looking strand in the middle here. So this is the bacterial chromosome. It's called a nucleoid in bacteria because it doesn't have its own separate nucleus like our DNA sits inside our nucleus. In a bacteria, it's just one big circular chromosome, and that just kind of folds up on itself. And that's what you see here as this massive spaghetti. And interestingly, for bacteria, they also have these extra little guys here. These are called plasmids. You can see a couple of them there in the cell. And those are extra chromosomal pieces of DNA. So you have the main bacterial genome, and then you have these extra little bits of DNA that go inside the cells. And those are very important for bacterial life for a lot of different species, and they carry some very interesting genes on them, and we'll come back to plasmids in a little bit. So that's bacteria in a nutshell. And there are, what I've got here on the screen is really just the tiniest tip of the iceberg in terms of bacterial species. There are just a ton of bacterial species, different types of bacteria. The ones I have on here are some of my favorite disease-causing bacteria. I know everyone has like a favorite disease, so I've just put a few up here of mine. And these ones are all very important to human health and have been over the years in many different ways. So we talk a lot in medicine about these. So down here in the bottom, this one that looks like a bunch of grapes, this is Staph aureus. And so this is one of these bacteria that is very common in infections in people. And it is one that also we see a lot of this antibiotic resistance in. So I'll get back to that. I love this one here on the far side, this Y. pestis. So Yersinia pestis, this is the bacteria that actually causes the plague. And so if you think back to sort of Middle Ages, human history, this was something that wiped out almost half of the European population back at that time. And so a very important bug in terms of human medicine and human health. And I often, so when I do similar presentations to this, we'll do them sometimes to high school classes and stuff like that to talk to some of the younger generation about the importance of using antibiotics properly. And so I'll throw up this slide and I'll ask people if they recognize anything. And of course, they always zoom right in on gonorrhea right in the middle there, and you get a few chuckles from the back of the class and stuff like that. But these sexually transmitted diseases, so things like gonorrhea and this T. pallidum over on the side here, this one that looks like a corkscrew kind of, that's what causes syphilis. And so these are very important bacteria in human health. They're spread around quite easily through the things that people do when people are being people. And for the over 60 population, there's actually some interesting pieces here. So diseases like gonorrhea and syphilis in the last about 20 years or so have actually been on the rise in the seniors population. And so there's a lot of talk about why that could be and what exactly people are doing that's causing that to go. And I'll let you guys imagine what that is. But the point of this slide is just to show you that there are a lot of different bacteria. And again, this just being a small smattering of them. And they come in all kinds of shapes and sizes and all kinds of weird appendages and stuff like that. And so what I want to do is contrast bacteria with viruses because they are different and they are very different. And so on this slide is, again, just a very small smattering of different types of viruses. These are, again, viruses that infect humans. And so you can see on the left there, so on the top line is kind of an artist's rendition of what the bacteria might, or sorry, the virus might look like. And then on the bottom is either, again, artist's rendition or actual pictures from like really high powered microscopes. So on the left side, there is the human immunodeficiency virus. So that's HIV, as we all know. And you can see that for the most part, what a virus is, to compare this to the bacteria. So a virus, it's got some sort of a shell, in this case, it's called a capsid. And then there's these lipid membrane on the outside for HIV. But it's some sort of a shell that surrounds the genetic material of the virus. And A lot of the ones I have on here are RNA viruses. So RNA, DNA, these are both different types of nucleic acid. So there's HIV, the one next to that, the rhinovirus. This is a class of viruses that cause like the common cold. If rhino you think nose and you get a stuffy nose when you get a cold, that's what these guys do. Same sort of idea though, you've got this capsid surrounding the genetic material on the inside. The influenza virus in the middle there is what causes the flu, similar type of structure. I don't know if anyone's heard of these ones yet. Coronavirus, there's one going around currently. And again, same type of idea. You've got this structure, this spherical structure that encloses the DNA on the inside. Varicella zoster virus, that causes chickenpox when you're a kid and shingles when you're older. And it is a very, very unpleasant disease if anyone has experienced the reactivation of the varicella zoster virus from within their body. So if you had chickenpox when you were a kid, you got to watch out for shingles when you're older. And they have vaccines and stuff like that for that now too. And then on the far right there is Ebola virus. So very different than these other ones in terms of structure, but kind of the same idea where it's got this capsid that surrounds it, and it's just the RNA on the inside. So those are viruses. They're much, much simpler. So they don't have any of these ribosomes and all the protein making machinery and stuff like that inside of them. And so because of that, a viral life cycle is kind of parasitic in nature, where they require a host in order for them to be able to replicate. So this slide here shows a virus, let's say it's a flu virus or something like that, comes in and this big brown blob is say one of the cells lining our respiratory system. So this virus comes in and it will make its way into our cell. So it uses those spike proteins that you've heard about on the outside to attach to our cell and gets inside. And once it's inside, it opens up and it releases its DNA or RNA, whatever the case is. And that goes into the nucleus within our cell, where all of our DNA is. And it will basically trick our cells into using our DNA replicating machinery to make more copies of the viral DNA. It uses our cells' ribosomes to make more copies of the viral proteins. And then those just get reassembled inside our cells. You can see here, so you've got the DNA being copied and you've got the proteins being made by reading that, and it reforms into that sphere, and then they burst out of our cells and we cough and sneeze them onto our friends and family members, and or not if we're wearing masks. But that's how they spread, right? And they get spread out in droplets and through the air. So that is the viral life cycle, and it's why, sort of strictly speaking, viruses aren't really living things, because they actually can't make copies of themselves. They require another host in order to do that. So it makes them very different from bacteria. They can't make copies of themselves. The viruses can't, bacteria can. They don't have all of those cellular structures that are required to do that, and so they have to infect a host in order to do that. So hopefully that makes sense. And I just want to take a minute now to chat about how important, and I showed you all those different bacteria and viruses. Throughout human history, these have been so important to human health and our livelihoods. And so what's showing here, this is data from the US, but they were looking at the mortality from infectious diseases in the USA in the year 1900 on the left, and then going towards 2010, the year 2000, on the right. And so you can see on the left that in 1900, the number of people who were dying from infectious diseases, and so you can see that rates per 100,000. So for pneumonia or influenza, there were 200 people per 100,000 who were dying from those causes. Tuberculosis, gastrointestinal infections, diphtheria, and then other infectious diseases. To the tune of 46% of the deaths in 1910 were from infectious diseases. So just to think about that, because I like to just pause on this one for a minute. Almost half of the people who died just over 100 years ago they died from infectious diseases. And when you compare that, you can see the lines as you move towards 2010 just plummet down. And that's 3% in 2010 of the population died from infectious diseases. So that is a huge change in that 100-year span for how many people are being killed, essentially dying from these infections. And so that's a huge change in human medicine. And if you, I like to put over top of this graph, if you look at life expectancy in that same time span, so in 1900, on average, if you made it to your 5th birthday, because childhood mortality was actually a really big issue back then as well, but if you made it to your 5th birthday, you could expect to live to about 45 years old. That was in 1900. And then just over 100 years later, that's up over 80 years in most countries around the world. And so it's again, just crazy to think that we've seen almost this doubling in life expectancy in kind of that 100-year span. And so what are the big reasons for this? And of course, there are lots of medical advances that have gone on in this in the last century. But one of the big reasons specifically related to infectious disease and why we've seen this change in what people are dying from is the discovery of antibiotics. So I'm a student of science. I love the history of science. So I'm going to tell you a little story about this man here. So this is Sir Alexander Fleming. Dr. Fleming was a bacteriologist. So he studied bacteria in the early 1900s in the UK. And what happened here, this, I guess I should mention, this is the guy who is credited with the discovery of our very first antibiotic, penicillin. So this is the man who discovered that and how that sort of worked. So he was working in his lab, set up some plates of bacteria. So you can see like this plate on the right here. This is a little petri dish, and inside it, it's got like this jello, basically, which is just a very nutrient-rich agar. So bacteria can grow, and they love that stuff. They eat it up, literally. And so he would plate bacteria. You kind of spread the bacteria out over that plate, and then the bacteria grow, and they form colonies, and that's what this kind of white stuff streaked across the plate is here. So those are the bacterial colonies. So he plated these things out, and then he went on vacation and came back to his lab, a week later, and realized that he had left one of the plates open. And there was, as you can see, like this, there was a fungal colony growing on it. And so rather than, tossing it out and saying, it's a failed experiment, he looks at that and goes, "Ah, isn't that interesting?" Because he was Scottish, and that's how I imagine he talked. And what he discovered, what he found was, hopefully you can see my mouse there, this circle, this circular area around the fungal colony, that white fuzzy fungal colony, where there is no bacterial growth. It's called like a zone of inhibition. So it's this area where the bacteria seem to be unable to grow, so they don't grow right up against that fungal colony. And basically what happened here was Dr. Fleming had stumbled upon this chemical warfare that's going on among microorganisms. So when you think about evolution and these bacteria and fungus that live, in the dirt and in ponds and things like that, everything still has to compete to survive. They need to get food, they need space to live, all that kind of stuff. And so animals, we do that with teeth and claws and nails, but bacteria and fungus don't have that. So they use chemicals. And so what we've got here is this penicillium colony of fungus is releasing into the environment a chemical, which we've called penicillin, that kills the bacteria. And so what that does is it allows that fungus room to grow. If it prevents anything else from getting at the food and the space to live that surrounds the colony, then it gives that colony more chance to go here and grow out and get that food and make more fungus. And then as it grows, it releases more penicillin and kills off the bacteria. So we really stumbled upon this warfare and just that's happening all the time amongst microorganisms, and discovered a way to turn that into a drug. So he was able to isolate the chemical, the penicillin from those colonies, and show that you could use this on people. And so it was in 1928 when he made that discovery, and through a number, over a decade of sort of refining that process of learning about the drug, the penicillin, and how to get it, make it in big batches and stuff like that. In 1941, it was first used. So there was a policeman in the UK named Albert Alexander who was out pruning his rose garden one day, and in an unfortunate accident, I don't grow roses myself, but I understand they are very sharp with the thorns, and he got poked with a thorn, I think in his face, and ended up with severe abscesses that were kind of behind his eyes and in his jaw and went down his neck and stuff like that. So these big infections of bacteria. And they treated him with penicillin. He was the first person to receive penicillin. And it actually reduced, they went down and he got better for a while. They ran out of penicillin because at that time, manufacturing this wasn't what it is today. And so they ran out, and it was before his infection was totally cured. And so it came back and he ended up dying, unfortunately. But the proof of principle was there that you can use these things to treat infections and cure infections, bacterial infections in people. So this was an amazing discovery. And of course, 1941, right during World War II. So a lot of production was going into, and work was going into getting enough drug produced so they could ship this to the front line. So soldiers who were ending up with, bullet wounds and all these horrific wounds from shrapnel and stuff like that would get infected. We were actually able to start saving a lot of these soldiers by providing them with antibiotics. And so there was this then explosion of discovery of different types of antibiotics throughout the middle of the 20th century. So you can see the timeline here, and each of those circles represents different groups, different types of antibiotics that have been found. So you've got the beta-lactams, things like penicillin and aminoglycosides, chloramphenicol, blah, 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 blah. All these are all names for the pharmacists in the world. But that's basically what we've got here is different types of antibiotics were being discovered. And it was, constant happening. People, every stone you turned over would be a new bacteria or a new fungus, and you could isolate some kind of antibiotic from it. So it was a great time to work in the antibiotic field, in the antibiotic discovery field. And so I just want to give a quick highlight then about how antibiotics work, because that will get us back to the main point of this talk. So if you think back to that bacteria that we had at the beginning, and as I said, we've got all these different types of antibiotics. And so different types of antibiotics work in different ways. So for example, those beta-lactams, like the penicillin that I talked about, they work by inhibiting cell wall synthesis. So they actually stop the bacteria from being able to produce a cell wall. And other antibiotics do the same thing, similar stuff with those guys. Other antibiotics will work by inhibiting the ability of the cell to produce proteins. So they stop those ribosomes from working and producing proteins. And some antibiotics will work by inhibiting DNA replication. So essentially what we get is, when I talked about protection and growth and information, those things that these bacteria have to do to stay alive, antibiotics work by blocking or inhibiting some of those functions from working. And so I hope now that this is starting to make the connection. If you didn't know this, and I don't think I mentioned, but antibiotics don't work on viruses. And this is exactly why, because viruses don't make their own cell walls, they don't have their own ribosomes, and they don't copy their own DNA. So if you have a viral infection, like a cold or a flu, and you get antibiotics, you're treated with antibiotics, not only are those antibiotics not doing anything to prevent or to get rid of the virus, then there are potential side effects that can come from antibiotic use that you are now being exposed to. So this brings us to this topic of antibiotic resistance. So we've got bacteria, that your run-of-the-mill bacteria, like these little guys down here, we toss antibiotics at them and they die. And that's, again, just looking at their insides. They've got their chromosome and maybe some plasmids. There's a fancy high-powered microscope view of a plasmid. But that's what those bacteria do. Now, antibiotic resistance is when you get bacteria that are resistant to the effects of those antibiotics. And there's nothing magical about this. This is literally evolution. So this is bacteria will acquire genes, changes in their genes that allow them to become resistant to the antibiotics. And this is just a natural progression of that evolutionary tug of war that's going on between the fungi and other bacteria that are producing these things and the ones that are trying to survive. So a fungus produces an antibiotic, and the bacteria say, they start to die, and then eventually a bacteria will become resistant to that, and they say, haha, and they can move in and fight and compete with the antibiotic or the fungus for space and food, and then maybe the fungus creates another antibiotic and it works better and kills off those bacteria. So this antibiotic resistance is something that's been going on as long as there are these bugs and fungi that are fighting with each other for space. So just to kind of take this point home, that this is just a genetic thing. So if I'm a bacteria, like this big brown cell here or yellowish cell, and these blue dots are the antibiotic that someone's trying to use to hurt me, then depending on what kind of mutations you have, there's different ways that you can become resistant to antibiotics. So if I have like this purple gene here, maybe that creates an efflux pump, which is, you can think of it like a little vacuum that as soon as the antibiotic gets inside the cell, that vacuum sucks it and spits it right back out of the cell. You could have maybe this orange gene right here that creates a modified cell wall protein, which prevents the antibiotic from even getting inside the cell to begin with, and so on and so forth. So there are, as many different antibiotics we have, there are that many different ways that bacteria can become resistant to those antibiotics. And so what does this mean for people? So one of the common misconceptions is that people are resistant to antibiotics, and it's not us. So it's not us that become resistant to the drug, it's the bacteria inside of us. So that's the inside of a person. It's all cleaned out so you can see what's going on. And inside of us, there are bacteria, and we've heard about, the microbiome and your good bacteria, and you watch the Activia commercials, right? With the lady dancing with the happy tummy because she's getting her probiotics. So there are good bacteria that live inside of us, and they help us digest and poop and fart and do all kinds of weird and wonderful things that human bodies do. And maybe you've one of those people who's unlucky enough that you've got some antibiotic resistant bacteria in there. So I just put a little plasmid inside these guys to denote that they have some antibiotic resistance. But for the most part, these guys are, it takes a little more effort to duplicate and make more bacteria when you've got this extra bit of DNA. And so these guys are outcompeted by all the good bacteria that are in our guts. So they don't grow as fast, and there's not as many of them. They don't really take over and do any harm. Now, the problem can come when you take an antibiotic, especially if you're taking an antibiotic and you've got a cold or a flu, right? Something where it's not going to help the virus. But what happens is that antibiotic goes in, you go in, you take it, it goes in our stomach, it dissolves, and it spreads throughout our system. And it kills indiscriminately any bacteria that are not resistant to it. And if you have a bacterial infection, that's fine. That's what we want the antibiotic to do. But the problem comes, especially as I said, if you're just going against a viral illness, when you're done taking that antibiotic. So the antibiotic's gone, and now those bacteria that have those resistance genes don't have to compete as much for space. And so you can start to get colonization and growth of these bacteria, so that, maybe the next time you have an infection, it's due to one of these guys, and they're harder to kill, and they require more or different types of antibiotics that can have even more severe side effects. So I hope this emphasizes why it's important to take antibiotics only when they're necessary. And again, this is just speaking to the antibiotic resistance piece. It doesn't even mention the other potential side effects of these medications. And they are medications just like any other drug you might take. And so they have side effects. And they can be minor, relatively minor, like an upset tummy, people get a rash, have a little bit of allergic reaction. But some antibiotics can have much more severe side effects. And to me, one of the most severe is this idea of antibiotic resistance, and that this is a problem we have to work on together. So as I said, this has been happening since the dawn of bacteria in this fight and this creation of antibiotics and development of antibiotic resistance. And so you can see here, this little timeline shows on the left-hand side when antibiotics were introduced, and on the right-hand side when we first started identifying antibiotic-resistant bacteria. So penicillin was introduced around 1941, 1943. By 1965, we were seeing these penicillin-resistant pneumococcal strains, and so on and so forth, to the point, some of them, like levofloxacin, which was introduced in 1996, as soon as it was introduced, we already knew that there were pneumococcal resistant or levofloxacin resistant pneumococci, all these words. And so we don't even necessarily get that sort of 10, 15, 20-year grace period where we can use the antibiotics without having to worry about immediately fighting off antibiotic resistant bacteria. And this is something that is further driven by our use of antibiotics. So this graph here shows along the bottom the months of the year, January through December. And what we're showing in blue is the number of prescriptions for penicillin. And So you can see January, February, March, it's flu season. Lots of people are going to the doctor with their coughs and colds, and they're getting prescribed lots of antibiotics. And then flu season goes away. It's the summer. We all go outside and spread around, and the number of prescriptions drops. And then as we get into August, September, school's back in, kids are doing what kids do and sharing germs and things at school and coughing all over each other. And flu season starts to kick in again, and we see those prescriptions go up. And so what they did in this study was compared this with the resistance to the penicillin in E. coli bacteria. And so you can see that when there's high use of antibiotics, we see a high amount of resistance in those bacteria. And as we use fewer antibiotics, that line indicating the amount of resistance drops. And then as we use more antibiotics, we see more resistance. So we know from studies like this that the more antibiotics we use, the more antibiotic resistance we're going to see. So that's why it makes it very important to use antibiotics appropriately and only when necessary, so we can help to reduce and limit the amount of antibiotic resistance we're seeing. Another problem that kind of factors into this is that this is what I showed before, right? This timeline of the 20th century and the development of antibiotics. And what I didn't show was after 1980, we hit what they've called the development void. So we kind of found all the easy pickings, and pharmaceutical companies stopped producing new antibiotics at the same rate that they were before. So we've seen fewer and fewer antibiotics being developed while we're using more and more antibiotics. So let's do that math. If we're seeing fewer antibiotics being developed and more antibiotics being used, what does that all work out to? The antibiotic apocalypse. So it's, it's a little tongue-in-cheek because obviously, it's not going to ruin the earth. It's just going to mean we'll go back to a point where people are dying from infections unnecessarily. But that's a very real scenario. And people have done studies on this. So to kind of put that, what does that mean, the antibiotic apocalypse? So there was a study in 2014 where they kind of did some math and looked at this. And they determined in that study that by the year 2050, we would be seeing more than 10 million deaths a year from antibiotic-resistant bacterial infections. And so what does that really mean? They had another little thing that I really liked here. So if you look currently, the causes of death. So antimicrobial resistance right now is about 700,000. So about 700,000 people a year around the world die from antimicrobial resistant infections. And you can compare that to measles and cholera and tetanus, so on and so forth, and cancer at the bottom there. So more than 8 million people a year die from cancer. Well, if we don't do anything to change the way we're using antibiotics and reduce our unnecessary and inappropriate use of antibiotics, then that 10 million number looks like that. So we would see more people dying from antimicrobial resistance than we currently see dying from cancer, which is scary to me because I think a lot of people know someone who's died from cancer. It's a very common disease. It's a disease of aging, and very few people, at least in my experience, can name a close contact who's died from antimicrobial resistance. And so what we're looking at here is seeing a switch in that and going back to kind of the dark ages of medicine, really. So if you think back to that graph that I showed in 1900, we had 46% of people dying from infectious diseases. So this orange line here, the orange line here represents that. And so in 1900, and you see the line go along, and then, Spanish flu in 1918, and then we continue on down, and it kind of bottoms out at some point in the 60s, and we're just seeing that reduction. So again, going from that 46% of deaths to 3% of deaths from infectious diseases. If we start to lose our ability to treat infections with antibiotics effectively, then we can imagine that line starts going back up the other way. And so people start dying from things that for about 100-year span were preventable. And that would be, I think, a record in terms of creating or developing and losing a medical miracle in really what is one human lifespan. So that also means we would be looking at a change in medicine. And when you think about all the different medical procedures that we have, so when we look at that rely on antibiotics, so when you think about organ transplants, one of the reasons we can do organ transplants successfully is because so in order to get someone to their body to accept an organ transplant, you really have to give their immune system a whack so that it allows that foreign tissue essentially to get integrated into their body. While their immune system has taken that hit, they're very susceptible to things like infections. And so we use antibiotics to help prevent those people from dying while they're waiting for their organ to sort of integrate into the body. Similar situation with cancer, right? So when people are treated for cancer, your immune system takes a big hit. And so we use a lot of antibiotics to help those people fight off infections while they're recovering from their cancer treatments. Surgeries, all kinds of surgeries, like it is considered relatively safe to be able to go and get a total hip replacement or a knee replacement, those kinds of things, because your chance of getting infection is low. The more antibiotic resistance we see, the chance of getting infection and the ability to treat the ones that do happen is low. And so as we see changes in the bacterial populations that mean more and more resistance, well, that means the potential for a bad outcome from a hip surgery or a knee surgery actually starts to increase, and people might have a sober second thought about whether or not they want to have that hip surgery. Things like sore throats. I mean, we can get to a point where sore throats can kill people again. When you have that strep throat, you get a bacterial infection in your throat. If your immune system isn't able to wipe it out and you don't have antibiotics to help kill off those bacteria, I mean, this is very close to your brain, and bad things start to happen when people can't clear out those types of infections. And, just to take it back to the policeman, Albert Alexander in the UK, like to go back to a point where, your hobbies, gardening, could potentially kill you is, I think, for me, a sobering thought and not a place where I think we want to be in medicine. So, with all the doom and gloom, I like to try and end on something positive. What can we do? We as people in the community and people who are getting healthcare, what can we do to try and be better stewards of antimicrobials? What can we do to make sure we're using these things appropriately? And so number one on my list is always don't request antibiotics when you're diagnosed with a viral illness. So if you go to your doctor and the doctor says it's a cold, it's a viral illness or a flu, they're self-limiting, which means they go away on their own. Your body just needs time to heal from them. And antibiotics won't help. So there's no need to pressure for that. And sometimes doctors feel that pressure where the patient says, well, I had my azithromycin last time and it went away. And then they feel like they have to do something to help their patient, and people end up getting unnecessary prescriptions. So having that conversation with your doctor or nurse practitioner, whoever your healthcare provider is, about whether or not antibiotics are required to treat your illness. Practice proper hand hygiene. So, I mean, again, being embroiled in the middle of a pandemic, I'm sure this is something everyone's heard a million times, but washing your hands to prevent yourself from getting sick in the first place means that you don't have to go to the doctor and risk potentially unnecessary antibiotic prescription. This is another big one, not sharing or using old antibiotic prescriptions. So, you get your antibiotic prescription for 10 days and you take 7 days worth and you keep those extra 3 pills in the medicine cabinet or wherever. Just so that next time you feel a little tickle in your throat, you're just going to pop a few of those pills. Please stop doing that, and don't give them to your neighbor or things like that. We had a patient partner that worked with us who had said one time, she makes the analogy of like, you wouldn't go and just start giving yourself chemotherapy to prevent cancer from coming. So why would you take an antibiotic if you think you've got a sore throat coming on, right? I mean, these are medications and they are prescribed by professionals for a reason. And then the last one is getting vaccinated against flu and other preventable diseases. COVID is a perfect example, causes an upper respiratory tract infection. So again, preventing yourself from getting sick means you're not going to go to the doctor and potentially get those unnecessary antibiotic prescriptions. So I will stop there. The whole idea here is that we need to ensure that we're using these things properly so we don't see the sunset on the antibiotic era. We do not want to get back to a place where we don't have the ability to treat bacterial infections in people. Because it means shortened life expectancies, it means people dying from preventable causes, and that's just not a place we want to get to in medicine. So I will thank you all for your attention, and I will stop sharing my screen so we can see everyone's